Hello, welcome to the Elephant DNA Lab. This is normally a wet lab that you would be doing in class. As we're virtual, we'll be doing it together online. We will be looking at the data and looking at a gel electrophoresis DNA lab that was previously run with 50 samples of DNA that represents DNA taken from ivory captured in a shipment. And you'll be determining where in Africa the DNA came from by matching the DNA to DNA from different populations taken from elephant dung, just like was done in the previous lab on the HHMI website. All right, so the first step is you are going to be given a DNA sample by your teacher. There are 50 samples total that are split up among 10 different gels. Okay. The introduction gives you a little background on the technique used to extract and how to run the gels. And in this box here, you are going to write down the number of the gel samples you are going to be analyzing. For example, maybe you're doing number 1 and number 2, and I'm actually going to be doing number 1 for you as an example, so I'm just going to say number 1 in this box for now. Okay, and so here you can see the picture of the gel. So the bright bands represent fragments of DNA that separated during the electrophoresis. So the current kind of pulled it out. The smaller fragments will be near the bottom, and the larger fragments will be near the top of the gel, near the wells, which are these holes at the top. On the left-hand side, in the first column, that is the DNA ladder. So the ladder is used to help you measure the size of the fragments. So these are all known size fragments. And underneath in the table, you can see here it says 1 kb DNA ladder. And so you can see the size of the band. So the top one here is 10,000 kilobase pairs. Okay. And then it goes to 8, 6, 5. So by counting the number of bands that you see and then lining them up with the band on the ladder, you can determine the size. And you can see here the first one has been done. So what you're going to do is count first. You're going to count how many bands you see. So 1, 2, 3, 4, 5, 6, 7. It's pretty faint down here at the bottom, but looks like 7. Okay, so count even the faint ones as well. This first band, now if you notice, it's higher than the first band on the ladder, which is, again, that 10,000 kilobase pairs. So because we don't know the size of it, we're just going to put that it's greater than 10,000 kilobases. So that's why for number 1, that's what it says. Okay, the second band lines up pretty evenly with the 10,000 kilobase line on the ladder. So 2, we just said it's the 10,000. Now some of these might be estimates, and that's okay. You're just trying to match it as best you can. Okay, so then the next line down would be about 8,000 kilobase pairs, and that looks like it kind of lines up with the third line on the first sample here. And so we went ahead and put 8,000, and then went ahead and did that with all of the rest of the bands all the way down. So you notice that they're getting smaller as you go down, the smallest one being about 600 kilobases. Okay, so once you've analyzed your band here, okay, so the next step is, if I go back to the assignment, your teacher will have created a class data sheet for you. So when you click on that, it will go to a Google Sheet that has a table with the lab results, which we don't have yet. So we first need to match our data with known samples. So we're going to first check the forest elephant database. So I'm going to click the link for that. Okay, and you can see here that it gives us the population ID, which is just a name that they give it, the park in which the population lives in, and what country that park is in. Okay, and if we skip over here to population STR, so that just is stands for short tandem repeats, which is the type of DNA fragment that we're measuring. And what we're doing here is we're looking to see if we can match our sample that we just collected data for. Oops, I'll go back here. Okay, with the ones that we see on the database. And the first thing I notice when I look at this is I don't see any that have 7 bands, right? And if I look here, these are all in order of the largest bands to smallest. These are the forest elephants, and it looks like the band sizes all start around 6,000. Here's one at 7,500 here. That's the largest, which is pretty far off from our data, which had a band that started at around 10,000, right? And then, so the first 3 bands are a lot larger. So that's telling me that, I don't think that this sample is from a forest elephant, but I need to check the other database to check and see. So the other database we have is the Savannah Elephant database. So now I'm going to click that. Oops. Go. Okay, and then right away, I'm looking here in the population STRs, and I can see, okay, these have a lot larger DNA fragments, and I'm seeing ones that have numbers that are closer. So this one here has 7 bands, although I'm only seeing 3 listed. The first one is bigger than 10,000, but then it goes to 3,000. So I think we can find a better match than that. Okay, and then here's one down here that has 1, 2, 3, about 7. 1, 2, 3, 4, 5, 6, 7. So it's got the right number. I see the first one is 23,000. Well, we don't know what the size of our first one is, but we know that it's greater than 10,000. So it could be that, right? This next one is 10,000. So we'll see what this one says. 9,400. That's close to 10,000. So that could be right. The next one is 6,700. And again, we have 5,000, but again, that's, if you're around about 1,000 kilobase pairs off, that's okay. It's, it should be within a certain range. If you're more than a few thousand kilobase pairs off, then that's probably not correct. It also makes a difference as you go down, though, too, down the list, because as you notice on the ladder, the amount between kilobase pairs gets smaller and smaller as you go down. All right, so where were we? So let's look at the 4th band. 2, 3, so 4,400. Okay, so now 5,000. All right, so far this is tracking. So of the different populations that I'm seeing, I think that this SS Boma is the closest match. So you can see here down below, that's what's been typed as the identifier, SS Boma National Park. Okay, and we could also include the fact that this is Savannah elephant. All right, so what we're going to do is copy that identifier and you can paste that in your class data table. Okay. And so you're going to have to wait for all of your classmates to fill this out. So you might be doing 1 or 2 bands depending upon how many people are in your class. So, then you would just move on to the next one and do the same with the next sample. And if you're working as a group, you can help each other out, kind of, check each other's work to see if, maybe there's some discrepancies. So definitely it's just good to kind of work with somebody on this. Okay, and please put your name up at the top here so that we know that who did the data analysis for that data. Okay, and you can see here, right, that each sample, some of them might look similar, so maybe those came from the same location. Some of them look different, so those maybe came from different locations. So every single sample, once you've collected data for all 50 and your class has completed the table, either as a class or maybe your teacher will go ahead and take this data and then summarize it in this second table over here and basically count up the number of samples you have from each location. And this is what will kind of give you an idea of where the poaching hotspots are happening. Are they happening mostly with forest elephants? Are they happening mostly with savanna elephants? And then within those species, which parks or countries, right, might have a higher occurrence of poaching and might need to have some resources to help stop that. Okay, well, I hope that helped with explain the lab for you, and good luck.